Mike Majerison here. Welcome to Top Med Talk. Thanks for downloading the podcast. If you like what you hear, do make sure you subscribe. This week, we're presenting to you our Xtreme Everest mini-series, which we streamed on the website to you live from Duke University in America on June the 14th and 15th of this year. For more details, all you need to do is search for Xtreme Everest Symposium, Human Tolerance of Hypoxia, Going to Extremes to Understand Critical Illness. And to find out more about this series, visit the website topmedtalk.com. This first piece is presented by Professor Mike Grocott. It's the first in the series and it's called Critical Care, Can We Learn from Environmental Physiology? Have a listen.
Top Med Talk.
Delight to be here and to, I guess, do the scene setting for the presentations that follow. And I have to say, so I'm particularly nervous today. The reason being, Top Med Talk are here, we're streaming worldwide. And just before I left, my mum rang up and said, I'm going to be logging into that thing that you sent me an email about. So, particular pressure. Hi, Mum. She also said, you've got to change that picture. It's completely fraudulent to have a picture of 10 years ago or more. It's all grey now. Anyway, what I'm going to talk about, I'm going to talk a little bit about oxygen and some of the basic physiology of oxygen, because I think it underpins many of the lectures that will follow today. I'm going to address this notion that environmental physiology, or more specifically using environmental perturbation of human integrative physiology, is potentially a valuable model for understanding both physiology and pathophysiology, and potentially particularly of relevance to critical care. And I'll talk about that in a general way, and then more specifically in relation to how we think about that in relation to the Xtreme Everest project, and then introduce the general structure of the studies that we've done, really to lay the way for the subsequent presenters to present some of the data and explain what we think we've found. So this is oxygen, my favourite molecule. It's a kind of geeky thing to say, but I've spent a fair chunk of my life being somewhat obsessed by this. This is oxygen in its liquid form at about minus 180 degrees C, which I think it's a kind of beautiful image. But as we all know, we typically encounter it in its gaseous form as a component of air in which it forms just under 21%. And it's what keeps us alive day to day. I'm going to divert a little bit because I think it is of relevance to a lot of what you'll hear today. This is proper history. In fact, this is way beyond history. This is geological time in relation to the history of the Earth. So on the horizontal axis, we've got 3.8 of the 4.5 billion years that the Earth has been in existence. And the red and green lines are the upper and lower bounds for estimates of the atmospheric concentration of oxygen. And you can see that for a very long time, there was essentially no oxygen in the atmosphere. And then over time, it increases. There's a pretty big bump, maybe a couple of 100,000 years ago, and it's now settled to a level that we're accustomed to, just under 20%. And there's a little question mark there, and that question mark is when we think oxygen first started to be produced on Earth through some very basic photosynthesis, and a consequence of that was this thing called a Great Oxygenation Event, or sometimes called the Oxygen Holocaust, which is probably the biggest single mass extinction event that's ever taken place on Earth in terms of the proportion of species that were wiped out. And that was a consequence of these cyanobacteria who had worked out, metaphorically speaking. They had became able to photosynthesize and therefore metabolize carbon dioxide and produce oxygen, and that oxygen was released into the atmosphere, and for a long, long period of time, the atmospheric level did not appreciably increase because it was absorbed in the various oxygen sinks, so it was absorbed in the sea, it was absorbed, for example, in surface iron deposits, and you can still see that in parts of Australia. And there came a point when those sinks were fully saturated and the oxygen level started to increase, and the vast majority of organisms that are around that were obligate anaerobes, so demanded to metabolize in the absence of oxygen, had a really bad time, and most of them became extinct. And the ones that didn't become extinct, a few of them got by in little ecological niches, but the ones that didn't developed a way of defending themselves against the toxicity of oxygen. So we always like to think of oxygen being a wonderful, good thing that keeps us all alive, but actually, way back in geological time, the ability to metabolize oxygen was something that kept you alive because it stopped you being poisoned. And those organisms are related to what we now call rickettsial bacteria, and they evolved, and you can see from that image, they're very reminiscent of a mitochondria, which is what they evolved into. And so we developed this balance in life, which was the plants happily photosynthesizing, producing oxygen that we then metabolize, expire carbon dioxide, which the plants metabolize, and that's the, if you like, the circle of life that keeps the biosphere of Earth going. But important that oxygen, although it's now producing energy, producing ATP, and is as an invaluable thing to keep us alive, originally it was defending against oxygen toxicity. That was why we developed these relationships with what are now the mitochondria. So there's this notion of the yin and yang of oxygen as a molecule. You can have carefully controlled, as we have in human cellular biology, carefully controlled metabolism and the production of energy, analogous to the internal combustion engine, or you can have uncontrolled oxygen chemistry, which can potentially be very harmful, analogous to this forest fire here. So what we like is normal metabolism. In this case, the example is carbohydrate. So controlled metabolism, the cellular biochemical mechanisms use carbohydrate through glycolysis, TCA cycle, and ultimately the mitochondria to produce ATP, which is what keeps us all alive, keeps the show on the road, underpins all our metabolic processes, allows us to move, reproduce, grow old, etc. And so this is fundamental to the basis of our life, our existence. But we also have, on an ongoing basis, continuing what we call oxidative stress, so the production of reactive oxygen species which are potentially hugely harmful. They damage lipids, they can damage proteins, they can damage DNA, if the production of reactive oxygen species is allowed to proceed in an uncontrolled manner. So it's critically important and of direct relevance to the physiology we're going to be talking about today that it progresses in a controlled manner, and then it's really important for metabolic signaling, so for normal pathways, the normal cellular biochemistry. I'm going to change tack now. This is one of the slides, it took me a long time to find it. It was one of the slides we used way back when we first developed the Centre for Altitude, Space and Extreme Environment Medicine at UCL. Kevin Fong and I set it up around the turn of the millennium, so 18 years ago now, with the benign support of Monty, as in he prevented other people from stopping us doing and was incredibly supportive financially and otherwise in allowing it to keep going. So our notion at the time, and this came from, we'd started teaching a bachelor's course, which became the most popular bachelor's course at UCL, around this notion that you could learn from environmental physiology, because perturbing physiological systems through environmental stresses allowed you to understand how they responded under stress, so if you challenge them, and also what their extremes of response were, because sadly in many of these environmental contexts the physiology can become overwhelmed and people can die. And we had Kevin Fong, who was a specialist in space medicine, Denny on board as a specialist in diving and hyperbaric medicine, a number of us interested in altitude medicine, and also individuals including Sandeep Dhillon and Chris Imray in the front row interested in thermal medicine, both at the extremes of heat and cold. And out of that, over a number of years, developed the Everest project. And the underpinning interest is really human integrative physiology. And although we're using environmental stressors, it is the use of human subjects and the use of integrative physiology, so the study of the biological function in an integrative manner, not in a reductionist manner. So not looking at simply what's going on in a cell or a cell preparation or a petri dish, but what's going on in the whole organ, the integrative physiology. And this graphic, I guess, is kind of useful in that respect. It's relatively straightforward to do experiments. I say that as a person who doesn't do experiments in the lab. but the ability to produce results and understand simple mechanisms in the lab is substantially more straightforward than it is in the whole human. But the problem is that the relevance, understanding what that means, because the conditions in the lab can be so contrived compared to what exists in the human. So, for example, doing oxygen studies, what level of oxygen do you study? Because we're not really sure what the level of oxygen at the mitochondria in normal human physiology is. So what are your controls? What are your interventions? So we have this payoff between experimental tractability and physiological relevance. And maybe there's a middle ground amongst the sort of the groups of cell cultures, or even more appealing, animal physiology. But actually history has taught us that middle ground is not the most productive. So this is a systematic review from the British Medical Journal looking at contexts where there have been studies both in animals and subsequent studies in humans, and the bottom line message is that we are often misled. We are commonly misled by studies in animals when we try to translate into humans. That doesn't mean we're always are misled, and there are some amazing discoveries that have come through that pathway. But it's often misleading, and particularly in critical care, we have some cardinal examples of molecules that were developed through animal experimentation. So this is activated protein C, the rather discomforting withdrawal of a very promising molecule about 7 years ago now, which is big news for the critical care community, and really just mirrored for me what happened when I was an intern when they withdrew the anti-endotoxin treatments. And there's been this history over decades of critical care interventions that were brilliant at treating rat and mouse sepsis, none of which have subsequently translated into the effective treatment of human sepsis. So this pathway is not the most productive for us. And there's, if you like, there's in some respects related and conceptually very important. One of the key things is that we now understand that the Cartesian notion that you can, if you understand the basic building blocks, you'll know what goes on in the whole organism, is clearly incorrect. The whole is substantially more than the sum of the parts, and we know that if just reading the genetic code, you cannot produce a human. You need all the molecular mechanisms that comes with cells and the combination of the egg and the sperm in order to effectively produce a human, and an identical genetic code at the start of that process can result in non-identical individuals, does result in non-identical individuals at the end because of all the other processes that come to play, including epigenetics. So actually the notion that we can understand everything from the petri dish or the cellular preparation is relatively unlikely. And if you're into human integrative physiology, environmental stimuli are one of the easiest ways to challenge human physiology, because humans are strange individuals. They voluntarily expose themselves to some fairly extraordinary circumstances, including altitude, and that provides a model for understanding perturbation of, in the case of altitude, of oxygen levels, but also of pressure in the case of diving, of heat in the case of people who choose to go to the poles, for example. So I'm going to move on now to the specific case of looking at oxygen and altitude. This, I'm sure, is very familiar to you, but just to put some numbers on it, horizontal axis is barometric pressure. The horizontal axis at the top is essentially 1/5 of that, so it's the partial pressure of oxygen. And the vertical axis is altitude in meters, and I apologize, this is in European, not American. At about 5,000 meters, so roughly 18,000 feet, which is around the height of highest human habitation, but also the height of Everest Base Camp, there's about a half the oxygen available that is at sea level. And at about 9,000 meters, so the top of Everest, 8,848 meters, 29,000 feet, there's about a third the oxygen availability that is at sea level. So 1/2 at base camp, 1/3 at the summit. And one of the important themes that will run through many of the talks is that, and this is a slide from a mentor of ours, Jim Millage, this notion that actually the amount of oxygen can be well maintained because of adaptation. So this is a cartoon of hemoglobin. If you think of the amount of hemoglobin as the width of these bars and the saturation as the shaded area, so you can have the same amount of oxygen being carried in the blood in more hemoglobin that is less saturated. I'll recapitulate that in our data. So this is some of our blood gas data. The white bars are PaO2, falling as you go from sea level up to nearly the summit of Everest. These bars are saturations, falling to a lesser degree. The hemoglobin level is jumping up, and so the darkest bars, which are oxygen content, are actually, in individuals who had time to adapt, are supernormal right up to about 7,000 meters, and it's only when you get over 8,000 meters that the oxygen content starts to fall. And it's important to keep that in mind when you hear the physiology we're going to go through. The levels of blood gases that can be tolerated are extraordinarily low, so this is some of our data very near the top of the mountain. There are 4 individuals here, the lowest of whom, who's sitting on the front row. In fact, two of the individuals who are measured, those little red stars, are sitting on the front row. The lowest of whom, in the beige jacket, Dan, had a PaO2 of 19 millimeters of mercury with completely normal physiological function. So talking, he was one of the 2 of us who took the blood gases, gases taken first time, talking fluently on the radio, walking around with a PaO2 of 19 millimeters of mercury. So it is extraordinary what is tolerable when individuals have had sufficient time to adapt. It's very rare in physiology. There's another example, one of very few that we could find. In clinical medicine, those sort of levels are always associated with serious pathology. So individuals who are pericardiac arrest or during cardiac arrest, there's a few case reports around profound opioid overdose, but non-associated with normal function. Another context in mammalian physiology is the diving seal, and you'd imagine that the diving seal evolves to understand at what point it should surface, and when the diving seal surfaces, it has a very similar level of PaO2. And another context is one that every single one of us has experienced, which is the human fetus in utero. And it is clear that around this level, so 19, 20, 21 millimeters of mercury, human development in utero progresses perfectly happily. So organogenesis takes place, the fetus develops normally. So within each of us, within all of us, is the capacity to develop and function normally with a profoundly low PaO2, but somehow in childhood and adult life we lose that capacity, but it's somewhere in our genetic makeup to recode for that. So the Everest Experiments. There were two reasons why we went to Everest to do this research. The first was what we believed, and it follows from some of the things I've said, but I'll go into it in a slightly more detail. We believed that we could understand something about critical illness, and particularly the relationship of oxygen to various aspects of critical illness from understanding the changes that you see with hypoxia at altitude. And the second was that we were not convinced by the established explanations of what happens at altitude at the time. So I'll take those one by one. This is a good friend of several people in the front row, a climber who became acutely ill with pancreatitis, and he's a great example of someone who had profound and sustained hypoxia. You can see he's got a tracheostomy, he had a severe acute lung injury, he ended up being filtered, all the usual paraphernalia that go with prolonged critical illness, and someone who had profound hypoxia for a long period of time and potentially would have benefited from the same mechanisms that we see in individuals who adapt well and are able to climb Everest, for example. And so we took the approach that if we studied healthy people on the right-hand side as you look at it, ascending to altitude where the main thing that was changing was oxygenation, that we could understand something from the critically ill where unfortunately are lots of things changing. They're a very heterogeneous population, different presenting complaints, different treatments, different responses to treatment. And I guess we're still on that journey, but I think we're feeling much more confident now that actually that will be a productive journey. There were some clues that encouraged us in this. One was that there are some recognised genetic associations between both the ability to thrive at altitude, so these were UK individuals who were elite high-altitude climbers, seemed to have an over-preponderance of a particular allele, the I allele of the ACE gene, so the angiotensin-converting enzyme gene, and that same allele appears to confer a survival benefit in ARDS, so an illness that is characterised by profound hypoxia. So that doesn't prove anything, but it's at least encouraging in terms of the inference that you can take information from one context and use it in another context. Well, if you look at the general pattern of physiological change that you see with acute exposure to hypoxia at altitude and acute exposure to critical illness, there are a lot of commonalities. So acute exposure typically results in a fight or flight type response, lots of catecholamines around, increased cardiac output, increased heart rate, whereas chronic exposure is much more akin to hibernation, and that's true in critical illness. This idea of multiple organ success, whereby metabolic downregulation seems to allow organs to tolerate the stress of a big inflammatory hit, and we have the same at altitude, where actually metabolic downregulation may be part of beneficial adaptation. And we know that if you aggressively increase oxygen delivery around the time of surgery with an acute injury, there's clinical benefit. If you do the same in established critical illness, it makes no difference, or indeed in some cases it's harmful. So some parallels in terms of the basic physiology. The second issue was that the physiological explanations at the time, which were all about, if you like, dealing with the oxygen cascade, so the notion that there's less inspired oxygen, in order to maintain adequate oxygen to the tissues, you need to squeeze the steps in between inspired oxygen and the tissues by, for example, increasing the amount of hemoglobin, increasing ventilation to increase the amount of oxygen in the alveolus, increasing capillarity to reduce the diffusion distance from any perfused vessel down to a metabolizing cell. And all those changes are true, they take place, but they don't explain beneficial or adverse adaptation. So if they were a full explanation of how you adapt well to high altitude, you expect that individuals who had extreme values for any of those variables, so the briskest hyperventilatory response, the greatest increase in hemoglobin, for example, would adapt best in terms of their ability to perform and tolerate hypoxia, and that's simply not true. And probably the most counterintuitive thing is that the ability to be an aerobic athlete, so aerobic fitness, which you'd think must be associated with the ability to tolerate hypoxia, because it's all about getting lots of oxygen down to metabolizing cells and running or swimming or whatever your activity is, chosen activity is, quickly. That is not associated with the ability to tolerate hypoxia. In fact, you'll see data today suggesting that actually inversely associated. So they're two completely different phenotypes, even though they're both about delivering and metabolizing oxygen. The best examples of that, so Reinhold Messner and Peter Habler, the first two individuals to climb Everest without supplemental oxygen, extraordinary high-altitude athletes. They were studied in great detail, so they achieved it in 1978. During the 80s, they were studied in great detail, and the really remarkable thing about them was how unremarkable their physiology was. So they didn't have a brisk hyperventilatory response. Their hemoglobin response wasn't particularly abnormal. They weren't that fit, counterintuitively, and yet they achieved this extraordinary goal. If you look at our data, and this is from a blood gas paper from 2007, the oxygen content, as I mentioned already, is well maintained, if not supernormal, up to very high altitude. So it's not simply oxygen content and oxygen delivery that's explaining the changes in performance. And so our notions, the hypotheses we brought into this experiment were based on the fact it's not all about oxygen delivery. It's not simply about getting oxygen to the cells. It's about something that's going on in the tissues. And in particular, we wanted to understand if it was to do with the microcirculation, so autoregulation and microcirculatory blood flow, or if it was to do with biochemical changes, so cellular metabolism and in particular mitochondrial metabolism, where the business end of oxygen metabolism takes place. This is the design. So we took healthy volunteers, the majority of whom were taken from the general community. They were trekkers who over 12 days trekked up to Everest Base Camp from an airfield relatively high in the mountains. I'll show you the geography in a moment. And they were experimented on in London before they left, and then sequentially as they ascended to altitude. And the key thing is that they ascended at exactly the same rate. So we planned this very carefully so that when you see differences in physiology, they're not about differences in how fast they went up the mountain. They are to do with their innate physiology. In 2007, we also had an investigator team who went higher on the mountain, and you'll see some of their physiology today. In 2013, the expedition that Dan Martin, front row, led, the design was the same, but the individual makeup was slightly different. So we can compare data between the two studies, but in 2013, we had nearly 70 Sherpas in one of the groups, compared with another group of lowlanders, so we were able to do comparisons between different racial groups. This is our laboratory. So sea level measurements before you go, a flight up to Lukla, around here, some baseline measurements in Kathmandu usually, measurements in Namche Bazaar at 3,500 metres, Ferrochet at 4,300 metres, and then Everest Base Camp at 5,300 metres, 18,000 feet. And then in 2007, measurements higher on the mountain as well. And what we now have is an extraordinary bioresource. So we have genetic, epigenetic, proteomic, transcriptomic, metabolomic, and physiological data on a huge number of subjects, approaching 500 subjects now, that we're attempting to integrate. And our challenge is now, instead of being about the logistics of expeditions and how to do this sort of science safely at altitude, are much more about complex data processing, the managing of large data sets, and the presentation and analysis of those complex interacting data sets. And this is really the notion that we have, that we can identify signatures of beneficial or adverse adaptation by combining these different physiological and biochemical and genetic signals, and through that do two things. Firstly, identify biomarkers that we can potentially use in the clinical environment, so markers of adverse adaptation, of beneficial adaptation, markers of susceptibility to adapting badly to hypoxia, for example. And secondly, to understand mechanism, because if we start to understand which biochemical molecules, which physiological changes are in play at any one time with beneficial adaptation, then we can start to understand whether it's the microcirculation, what it is that's affecting the microcirculation, what it is that might be driving changes in mitochondrial function, and thereby develop novel interventions and then empirically test those. And those are the things that are potential treatments over time in humans. And amongst other things, we have clinical trials at the moment going on looking at different levels of oxygen, so the notion of permissive hypoxemia, tighter control of hypoxia, of oxygen therapy, avoidance of hyperoxia in both the perioperative and the critical care setting, and trying to understand the biomarkers that predict susceptibility to not tolerating either hyperoxia or hypoxia well, and can be used to track whether that is successful or not. But of course, the holy grail at the end of it is developing novel therapies that essentially mimic beneficial adaptation, if you like, mimic the Sherpa phenotype. So I'm going to leave it there. Only to go back to this slide because it's one of the things that really gives us hope that this will be a successful journey. This fact that all of us, each of us, have within our own physiology somewhere the capacity to survive and thrive in profound hypoxia. If we can just work out what those changes are, we potentially have some very valuable treatments for patients. Thank you very much for listening.
Mike, Thank you very much indeed. We're going to take questions at the end, but just while we change over the technology, Mike, I'm just going to press you on one thing, which is a recurring theme. We've sort of alluded to it already, is there's a beautiful set of chambers over here, just around the corner, where you can make people hypoxic. Couldn't you have just done all of this in a chamber?
Yes and no.
Right, can we go home now?
Yes and no. So you can do much of this in a chamber. There are subtle differences. So we and others have done work. The changes that you see in the same conditions in a chamber and at altitude are subtly different for reasons we don't fully understand. But they're not different to a level that is material. So you can effectively mimic the physiological response to altitude in a chamber. But I don't think you can persuade 200, well now nearly 500 volunteers to sit in a chamber for 3 weeks. The pleasure factor is pretty low, the motivation factor is low. So I think there's a simple pragmatic notion from that respect, and it's more expensive. I mean, if you think of the chamber time we would require to redo just the 2007 experiment, it would completely overwhelm.
So just to be clear for everyone, the volunteers took 3 weeks holiday. They paid to go on that holiday. And then they raised money to give to you, us, to be guinea pigs for our experiment. And you Absolutely. Don't think they'd sit in the chamber over the road under the same model?
It's a judgment thing, but it seems unlikely that they would both contribute money and volunteer to use their 3-week Easter holiday to come and sit in. The delightful as the chamber is. But
well, let's try it at some stage.
We can test it.
Top Med Talk.
Professor Mike Grocott there, an excellent first presentation. So the next presentation will follow tomorrow. And don't forget, you can meet the Top Med Talk team. All you need to do is get to the EBPOM website, ebpom.org/meetings. Our next big event is on the 28th through to the 30th of September 2018. EBPOM USA, the Chicago Masters Course of Perioperative Care Practicum. I'll see you there. ebpom.org/meetings. And for more information on this podcast series, get to our website, topmedtalk.com.